Let's consider figure 9.16 in our book on page 171. We know that plants that have a seed color that's either yellow or green consist of having alleles big Y and little y, with the big Y allele conferring the dominant form, which would be yellow, and the little y allele conferring the recessive form, which is green. So any genotype which consists of this big Y, little y, or big Y, or little y, those are all the genotypes. That's going to confer the phenotype, which is what we actually visualize. So a gene might consist of a big Y, little y genotype, but the phenotype that results from the expression of this gene is going to be yellow because we know it has the dominant allele, large Y. If it were little y, we would know that it was recessive, and therefore the seed would be green. So in considering this example in our book in figure 9.16, we also have round versus wrinkled in seeds. So we have an additional gene here. So the additional gene, big R, little r, would confer that the seed is round. Now if we had little r, we would end up seeing wrinkled seeds, as is shown in the book. Now if we had big R, because both of these alleles, R and R, are the large dominant form, we also know that these are going to be round seeds. So let's consider the cross that is in the book figure here. And in this book figure, we have yellow round seeds, and according to the very top of that figure, these plants are large R, big Y, little y, and we're going to cross two plants like this. So we know that the other plant is also going to have the same genotype. So in doing that, we want to look at what are the possible outcomes from a cross like this. So what sort of seeds are we going to have representing? And to do this, we want to construct a Punnett square. Okay, so here we have a blank Punnett square. So what do we need above each one of these boxes is are the possible alleles to be crossed. So since we know that both individuals have this genotype, I'll write it out here, then we know that we need to have one R and one Y per box. So here we want to look at what are the possible configurations of R and Y. And so we know that we could have a large R and a large Y, so that'll be in our first box. We know that we can have a large R and a little y, and that'll be our second box. We know that we can have a little r and large y, and then we know we have a little r and a little y. And we know that because this is the first individual, and the second individual has the same genotype as the first, then we know we're going to see the same possibilities down this side. There we go, I got those backwards, my apologies. Okay, so we have large R, large Y, and so what we see here is we have this cross here. And this cross is going to tell us, okay, so in this first box, this one here, we're crossing R and Y by R and Y, so we know that the resulting offspring are going to have two large R alleles and two large Y alleles. So this first individual is going to have the genotype big R, big Y, big Y. The second individual, again, we've got two large R's and a large Y and a little y. And then the next one here, we go to these two. We have large R, little r, large Y, and then large R, large Y, little y. So that's our first row there. In the second row, we're going to look at this individual and these four columns again. So we have big R, and this time we have big Y from this on the top row, or top column here, and little y from right over here. So then we have big R again, and now we have two little y's. A big R and a little r from up here, and a big Y again and a little y. And then big R, little y, little y. So if we consider this third row, now we have, and let's just go ahead and make this the same as the other one so we don't get all confused here. Little r, big Y. There we go. And we're going to do the crossing then up here. So we have big R, because we have a little r over here, and big Y, big R again from this column, and little r from over here on the left, and then big Y and little y. And then we have two little r's, a big Y and a little y, and two little r's, and a big Y and a little y. So this one here, you'll notice if you were watching closely, I actually made a mistake here in this box, because we don't have a little y on the left here. We have a large Y, and we have a large Y up here. So this one's actually large Y, large Y. Okay, and then this final one, we have little r and little y, and we go through again here. Perhaps this lettering scheme wasn't the best in retrospect, but make do with me here. And two little y's, little r, big Y, little y, and little r, and little y. So now we've determined all of our possible genotypes that would result from the cross between these two individuals here in the corner. Now we need to figure out what are the ratios. So we go through and we count how many of each type of these genotypes we have. So in this first one, we only have one big R, big Y, big Y. So we go like this. There we go. Now we have one. Now if we go through, we can look and we can see that here are two like this, big R, big Y, little y. And so we have two like that. And we have one big R, and one little y, little y. Now this is going to be your genotypic ratio. So if we look over here again, now we see that we have a big R, big Y here, a big R, big Y here. Where else do we have one? Those are the only two. So we have two big R, big Y. Okay. And then we have four big R, big Y, little y. And those are the ones that are coming through right up this middle. So you may have noticed that we have a pattern here that's emerged. And this may be true, but more often than not, it's the result of the kind of cross that we're doing where we have two individuals that have the same genotype. So we know that on the other side of this middle part, we're going to have a 1, 2, 1, 2 pattern. And these are all, again, your genotypes. So you would go through and you would have one little r, big Y, two big R, little y, Right here, you have one little r, little y, and two little r, big Y, little y. Okay, so now we've just determined our genotypic ratio. But now each one of these, in turn, confers some sort of phenotype, which we know to be either the round or wrinkled C type or the yellow or green C type. Okay, so I've put this in here so it's a little bit easier to read. So what we want to do now is determine our phenotypic ratio. So we know our genotypic ratio is 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. So phenotype then is what the genotype is conferring, what we're actually visualizing. So in this case, we know that the large R is going to be indicating that you have a round seed, and the little r is wrinkled. Okay. There we go. We also know that the large Y is a yellow seed, and the little y is a green seed. And we know that large R and large Y are always dominant over little r and little y. So any time where we have, for example, this large R, little r, we know that we're going to find a round seed because the large R is the dominant allele and therefore it is the phenotype that we see. So in looking at our ratio here, we know that this first one is going to be round and yellow because we have a dominant R, the large R is round, large Y is yellow. So we know we have at least one round and yellow. We know that this next one here is also round and yellow because again we have the dominant two large R's, and we have one large Y and one little y, indicating that the large Y is dominant to the little y, the recessive. We know that here we have another round yellow, or another two round yellow rather, because we have big R, little r, big Y, big Y. Here we have four round and yellow seeds because we have the big R, big Y, little y. So in counting this up, we have 1 plus 2 is 3, plus 2 is 5, plus 4 is 9. So we have 9 round and yellow. Okay, the next one would be looking here because in this column, this one is different. So we know we have a round and green because we have two little y's which confer the recessive here. So we have one round and green, and we see that we have another two round green over here because we have again the dominant R and the two recessive y's. So in adding that up, we have 3 round and green. If we look at this next one here, we see 2 little r's and 2 big y's. So we know that this one's going to be wrinkled and yellow. And we see here 2 little r's, meaning wrinkled, and 1 large Y and 1 little y, indicating, again, that this is going to be yellow. So adding those two together, we have 3 wrinkled yellow. And then finally we're left with one other type. So we have 1 wrinkled and green. So now we have our phenotypic ratio here, 9 to 3 to 3 to 1, where we have 9 round and yellow, 3 round and green, 3 wrinkled and yellow, and 1 wrinkled and green. And this is what we would expect to see roughly of the ratio from a cross between two individuals of this big R, big Y, little y cross. So over here, we have our genotypic ratio, the 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. And over here we have our phenotypic ratio, the 9 to 3 to 3 to 1. And this is how you generate a Punnett square and how you determine your genotypic here and your phenotypic ratios here.